So my name is Johnny. I'm going to be presenting a project that came out of a master's thesis I did last year at Imperial College London, together with some collaborators at Kew Gardens. So plants and humans have co-evolved through all of our shared history. Plants provide food, materials, and a whole range of sociocultural uses. Of the about 340,000 currently accepted plant species, about 10% have some record of use. And now with worldwide declines in ethnobotanical knowledge and rapid biodiversity loss, it's an important time to catalog and understand and take advantage of this information. But previous ethnobotanical studies and surveys are narrow in focus and often only look at certain clades or geographic regions or cultural groups. So this means that the phylogenetic distribution of useful plants has not been investigated at a whole tree scale, which leaves potentially many useful plant species, the uses of which are unknown or not yet recorded. And filling in these gaps could contribute knowledge that could help inform sustainable development or direct conservation efforts. So to try and get a better understanding of those gaps in plant use records, I began by looking at phylogenetic patterns of plant use. This question formed the bulk of a previous project. And in that study, which I don't have time to go into in depth right now, I used the World Checklist of Useful Plants, which is the largest data set available on plant use records, and a land plant megaphylogeny to look for patterns of phylogenetic clustering and phylogenetic signal of useful plants in these 10 use categories: animal food and environmental uses, which includes things like fertilizers and ornamentals; gene sources, which are crop wild relatives; fuels, human food, invertebrate food; materials, medicines, poisons; and social uses, which are like smoking materials or religious charms, for example. So the key takeaway from that was that all use categories showed some degree of phylogenetic signal. This is a family level tree with colored squares indicating a species with a given use in a family. And even with that simple measure, you can see the way that uses may be distributed. Here, highlighting some diversely used families and some less diversely used families. So once I had that information, the evidence of phylogenetic signal suggested that the distribution of uses could be modeled. So next, I thought about what potential predictors of plant use could be incorporated into a model and what that might tell me about the drivers of use. And then with the predictive power of that model, combining a wide range of factors, what predictions could be made about unassessed plant species? And what could those predictions tell us about underexplored or undocumented clades or use categories or regions? So to start thinking about how to model and predict plant use, I began by trying to assemble a diverse set of potential predictors that could capture the range of factors that make a plant useful. Most previous studies that modeled and predicted plant use focused on phylogenetic signal of, for example, specific trait measurements like alkaloid concentration. But that's only part of the story. Plant use is part of a multifaceted relationship connecting plant evolution within the environment, but also plant encounters and relationships with humans. And so I wanted to somehow account for these factors as well. So altogether, the potential predictors that I was working with fell roughly under these three groupings. I'll start by discussing the phylogenetic predictors. The trees that I used to derive these were complete megaphylogenies of all angiosperm and gymnosperm species in the World Checklist of Vascular Plants. To build these trees, we started with a backbone of 75,000 species, all represented by molecular data. And then the species that were not present in this backbone tree were added by binding a new species tip with a random branch length to a genus node in the backbone. And then this was repeated 100 times to generate 100 megaphylogenies each for angiosperms and gymnosperms. And these 100 tree sets will be used for the modeling to capture a range of phylogenetic uncertainty. But today I'm just presenting on the output of a single tree chosen from the set. So now I'll go through the phylogenetic predictor variables that I use taken from those megaphylogenies. First, the total branch length, which is a sum of the branch lengths of all species with a given use in a genus. The nearest neighbor distance is a measure of phylogenetic relatedness. It's the distance from a species in question to its closest relative with a given use. Intensity is a measure of the number of species with a use in a genus. Diversity is the average number of uses per useful species in a genus. And the evolutionary distinctiveness to look at traits that may be found in evolutionarily distinct lineages. The ecological and cultural factors I collected from a few different data sets. The ecological data came from the World Checklist of Vascular Plants, and the use records from the World Checklist of Useful Plants, as I mentioned. In the World Checklist of Vascular Plants, plants were sorted into more than 250 somewhat ambiguous life form categories. So these were condensed into these four more broad categories that have been used in previous studies. The biome data sorted plants into these 7 biomes. And I also incorporated the interactions of use categories by preserving off-target labels. So for example, a plant's use as a poison was treated as a predictor for its use as a medicine, and vice versa for all the other uses. I also included distributions for both native and introduced ranges. A history of human introduction can be evidence of a plant having some use. But there's also you can say, cultural distribution through human space. And for one measure of this, I got the number of languages spoken across the distribution of each species. And I retrieved the number of page views for every plant species on Wikipedia for the 6 most spoken languages and on the Chinese language equivalent by Baidu. So altogether, there are roughly 240,000 species for which I have a complete set of 29 variables for use in the modeling. So for the modeling, I went through a few steps. First, I checked for collinearity amongst the numeric variables. No collinearity issues were found. And then I took 4 different decision tree-based machine learning classifiers, as these work well with the diverse predictors like what I was working with, and 3 different training and test data partitioning schemes, and compared those in a 10-fold cross-validation to choose the best combination of data partitioning scheme and classifier. Brief, these decision-based classifiers work by combining thousands of decision trees generated from the training data into a forest that then collectively votes on the classification of new data introduced to the model. Then with the ideal model and data partitioning scheme, I performed hyperparameter tuning for these 3 hyperparameters, which are things like the number of trees in the forest and the number of splits at every decision node. And then one model was made, trained, and tested for each use category, and the ideal hyperparameters were selected independently for each model. So this is just one of the model performance measures that I calculated. The plot here shows the average classification error across the 10-fold cross-validation sets. Medicines is the largest category of useful plants by far, which makes it harder to predict for. And social uses are also strongly affected by cultural and human preferences, which also makes prediction a challenge. But across the 10 validation sets, the performances were pretty consistent. The classification errors were low, even though there was a little bit of range in some of them, showing that the models were performing well in the testing and validation. One of the really useful aspects of the decision tree models is the ability to measure the importance of a predictor and making the classifications. Across the board, phylogeny is consistently the most important variable. There are a lot of really interesting interactions here to talk and think about, but I'm just going to point out a few. Material and environmental use are important predictors for fuel, which is something that might be expected considering a trait like woodiness. Poisons and medicines have a lot of overlap, again, in line with expectations. And lastly, social uses show an association with languages, meaning a lot of potential for human encounter, but also with human food and medicine. And those are groups of uses with a lot of overlap, if you think of like tobacco or ginseng. So once those models had been trained and the performance is assessed, I used them to make predictions across the remaining roughly 200,000 plant species with no record of use. Plotted on a family level phylogeny here, newly predicted useful species were found in almost all families with a distribution across the tree similar to that of known useful species. With some families, like the orchids here, having many more predicted useful species in the bright color than known. In comparing known and predicted useful species by use category, here I've plotted the percent of known in the dark color and predicted in the bright color species in each category. So again, materials and medicines dominated as the largest categories, but it's also interesting to see a lot of potential new predicted species in the poisons, social uses, invertebrate food, and animal food. Coming back to life form and biome, I was most interested in looking at the relative potential for new species. So to get an idea of the potential, I compared the proportions of predicted useful species in each life form category with the proportion of known useful species in each life form category. And then I plotted the differences here, which can kind of show you which uses are overrepresented in each life form in the predictions. Overall, the epiphytes are the smallest category, so it wouldn't be expected to see many known or predicted useful species, but it's interesting to see that environmental uses and social uses in the orange and gray have a greater proportion of predicted species than expected. Woody perennials have a lot of known material use species, and here we can see a lot of potential for new material species in that life form category as well. And lastly, the biome differences are quite interesting, I think, as they capture an interplay of biodiversity by biome, human-plant interactions, and ethnobotanical survey completeness. Here again, I'm showing which uses are overrepresented in each biome. Medicines in the subalpine and subarctic biomes sticks out pretty clearly. This could possibly be a consequence of low human population, that these areas are understudied, or areas where traditional ethnobotanical knowledge has been lost. There's also very high potential for animal use species in the desert and dry shrubland biome, which could have implications for agriculture and farming under increasing desertification as a result of climate change. So to wrap things up, a couple words on limitations. As mentioned, there's quite a bit of phylogenetic uncertainty due to the lack of megaphylogenies fully supported by molecular data. We hope to account for this using the 100-tree hypothesis sets that are mentioned. And while the data are quite coarse, working at this whole tree scale, that's the only data that's available, but also makes an interesting challenge to try and quantify all the cultural and qualitative factors that inform plant use. So these predictions are also not necessarily informative on a species by species basis, but instead can highlight clades, areas, or biomes, et cetera, for deeper ethnobotanical exploration that can then inform things like conservation and sustainable development efforts. The interactions and relationships between the drivers of plant use also add to our understanding of humanity's relationships with plants. It's a relationship that goes beyond use and exploitation, and it's part of the story of our co-evolution and shared history. Lastly, I'd like to thank my supervisor, Sam Piernon, and other collaborators, Maribel and Felix Forrest at Kew Gardens for all their help and advice on this project. Thank you for listening.